Hi, I'm James Verdier, and welcome to the American Institute of Biological Sciences BioScience Talks, which is a forum for integrating the life sciences. On the second Wednesday of each month, we discuss the latest bioscience publications. And as a reminder, if you'd like to read more, point your browser to academic.oup.com/bioscience. Today, I'm joined by Dr. Andrew Hope, who's a research assistant professor at Kansas State University's Division of Biology. He joined me to talk about natural history collections, and more specifically, the effects of specimen removal on wild populations. So by looking at some older data on small mammal collections in particular, he and his colleagues were able to tell whether there was any discernible effect on a broader group of animals. But I'll let him describe those results. So let's get straight to it. Dr. Hope, thank you very much for joining me today.
I am happy to speak with you.
Okay, just before we get started and into the contents of your article, I was hoping you could tell me a little bit about natural history collections and the field in general. You know, why are samples and specimens taken, and why are they kept?
Well, natural history collections have been an integral part of understanding biodiversity since we started looking around and trying to understand the dynamics of how species form and relate to one another and how they go extinct by extension. For instance, Charles Darwin and much of his research in understanding evolution was based on specimen archives that he collected on his journeys around the world. And so really it's this concept of forming fundamental biological baselines from which we can begin to answer all kinds of questions depending on our different scientific fields. And so these historical baselines are really important for maintaining evidence of what species occur where at particular times through the past.
And where are these collections housed? You know, are they at a university, in a basement somewhere, museums, that kind of thing?
Yes, generally speaking, the best place to house these specimens is in public research archives, and those can either be part of, say, an academic institution or part of, say, a state or a countrywide museum. And so the goal here is to maintain these collections in a public manner so that both researchers and the public and educators can use specimens going forward. And so they should all be cataloged in a way to maximize their use.
And for these specimens, is this something where you can kind of collect once, or is it a situation in which you need to continually renew the collection with new specimens?
Yeah, it really depends on the question. When I go out into the field myself, I generally perform what's called general collecting, where you're setting out traps that capture a representative cross-section of the biodiversity that occurs in a given area. And so generally that means you'll get more specimens of the more common species and fewer specimens of the rarer species that occur in a given area. It also depends on the kind of discipline that you're working in. If I'm an evolutionary biologist, and so I'm interested in going out and collecting specimens across a landscape to understand how environmental change has influenced their evolution and genetic diversity through time. And so it consists of going out and opportunistically sampling to maximize the geographic scope of your species of interest, what the taxa that you're trying to collect. Whereas, for instance, long-term research ventures that are looking at an experimental setting in a much more refined area will have the potential to collect samples repeatedly in a single place.
And so, you know, in this case, you could have a situation in which you're not only looking to see, you know, the species abundance or range, but also perhaps its change over time.
Yes, absolutely. I mean, one of the biggest values of having these collections is that we can go back in time and use the information that's stored in these specimens to compare the situation from the past to the present situation and how that's changed through time. And so optimally, we would have repetitive sampling from any given area to build this time series of specimens that we can use to look at change.
And, obviously there are some obstacles to performing this kind of specimen collection. What are some of those obstacles? There's obviously the effort, but kind of what are some of the other challenges associated with this venture?
Yeah, the interesting part of collecting opportunistically is that many of the places that we'd like to try and fill gaps in and collect specimens from that haven't been sampled before are very difficult places to get to. And so there's a lot of both financial and time investments in these field collection efforts. And so really part of the benefits of collecting these specimens in their totality, including associated biodiversity, which I can talk more about, is an ethical responsibility to make the most of those efforts. And so you're not just going out and taking a small amount of samples for your own particular research, but you're collecting as much information as you can to provide so that other people don't have to go out and repeat your efforts into the future.
And that would potentially allow you to have less of an effect on, populations in the wild.
Yes. And so part of this study that we can talk about more is to try and examine what kind of effect we actually do have as scientists. And really these sampling efforts are a minuscule impact overall on natural populations compared with other both natural processes and other human-related processes that are occurring.
So in assessing the effect on, wild populations of sampling, how do you begin to address that question of how big an impact you're having?
Yeah, so this was it's not an easy thing to do. And the reason is because generally speaking, the best way to address the relative impact of specimen collecting is through an experimental setting where you're setting up an area that has specimen collecting and an area in proximity to it that does not. And so you're basically doing a statistical comparison of the impacts of collecting. And generally speaking, those kinds of experimental settings are done within the fields of, for instance, ecology, where specimen collecting is not often the major focus of those studies, whereas with evolutionary biologists and biodiversity scientists that fundamentally rely on these specimen collections to perform their research, it's very often not within an experimental setting. And so there's not many instances where the relative impact of these collections has been rigorously has been rigorously quantified, I should say, statistically.
And so how did you address that particular problem in this study?
And so in this study, I was fortunate basically to find a long-term data set based on a past ecological study associated with a long-term ecological research site, which was in the southwestern U.S., that contained this kind of side-by-side comparison. And it wasn't an original intention of the field efforts that occurred, but it provided a great opportunity to statistically assess the impacts of these. And so we had a situation where you had field crews out there performing mark and release sampling, where the animals were caught and tagged, say, with an ear tag, and then released. And this was done repeatedly, multiple times a year over 7 or 8 different years. And next to those sites, you had exactly the same kind of site where animals were collected and euthanized and processed to produce museum-grade specimens for the express purpose of looking at their parasite faunas. And so this provided a means of statistically comparing these two different collection techniques.
And just for our listeners who have yet to read the article, what were the species that were sampled?
So this study was sampling small mammals. These are vertebrates that include both rodents and shrews. And so basically you're looking at mammals from, say, the size of a small ground squirrel down to the size of a very small shrew.
Now, when they originally performed this experiment, Were they aiming to collect this sort of data, or is it just a happy accident that they wound up doing it?
It is actually it's a happy accident, but it illustrates very well the values of performing specimen collection, at least to a certain extent, particularly within these long-term studies. And so you have both a set of sites that you continue to do mark and release sampling, and so you can get long-term population variability data, but at the same time you preserve specimens that can be used to answer a whole other suite of questions related to, say, the evolution of the species, changes in the distribution of different genetic populations through time, and also coevolutionary dynamics of the mammals and their parasites, for instance.
Okay, so you're able to collect both of those kinds of information. Were these done at similar sites? Is there any reason why they didn't do, both the capture and release and the specimen collection at the same sites? Or
Yeah, they were basically, the sites were in proximity, and so within about 500 meters of one another. So very close together. And so basically you had sampling webs, which were basically a radial array of traps for the mark and release, and then right next to them you had another set of webs where specimens were removed. And so the aim was to maintain long-term continuity in understanding the population dynamics from the mark release sites without removing specimens, and then to maintain continuity in knowledge of variability in the parasite faunas from the removal sites.
Okay, and did you find any significant differences in the effects on populations or anything else from doing the mark release versus the removal?
Yeah, so the point of this study was basically to provide an opportunity to statistically test the impacts of specimen removal, and the way this study was set up allowed for that. And so the results of our analyses indicated that there were no real significant differences between the removal sites versus the sites where specimens were marked and released afterwards. And so it really shows that, well, it showed two things. First of all, that the impacts of scientific removal were minimal and not significant. And at the same time, it showed that populations did, however, fluctuate fairly dramatically. But those fluctuations were not in response to these scientific collection efforts. They were more related to other more influential variables such as climate dynamics.
So you don't see any patterns such as, an initial drop in population after the sampling effort begins, and then a maintained low level or declining rate?
Yeah, that would be one of the expectations, that you would see an initial drop, maybe with a rebound in populations after the removal, or you might see a steady decline in numbers over the course of a number of different years. But none of these patterns were detected throughout multiple years of sampling and over multiple different sites.
So the takeaway from that is that you can do some quantity of removal sampling in some settings without having a negative effect on the wider population.
Yes, absolutely, and I think this is the big take home message here is that the amount of specimens that we as scientists remove from the landscape really has no. It's not a big enough effort to have any significant impact on natural populations compared with many of the more natural phenomenon that these populations are dealing with, such as climate change or such as predation by predators such as foxes, for instance, which might eat several thousand mice in a year for a single fox. So really, it's an effort to try and put in perspective the relative impacts of scientific collections versus the relative benefits that those specimens provide.
So could you tell us a little bit about some of the benefits, that you get from this type of sampling effort versus a capture and release type?
Yes, there's numerous benefits, and hopefully I can get through some of the more important ones here. One would be just to verify that the specimen that you've caught is exactly what you think it is. And a lot of the time, what we're dealing with, I work with small mammals. When you're out there on a landscape capturing these specimens, they may look very similar to a bunch of other different species that you may find in the same area. And so having a specimen in hand allows you to verify that what you're calling something is actually what it is. And that's just a baseline reason. One of the other major reasons I would say is of having specimens through time is to allow for repeatability in scientific inquiry. And this is one of the major issues with not collecting specimens for research that relies on specimens. And so it's possible, for instance, to go out and take a small blood sample from a bunch of specimens and use that for a genetic analysis. But what you can't do is then go back and repeat that study after it's been published. And repeatability and falsifiability in science is a really critical part of moving forward. And so having these specimens in long-term archives basically allows the same scientist or anybody else to go back and repeat that study and repeat it in a different way or in the same way, just to make sure that the results were the same.
That speaks to some of the major benefits. And we've established that, at least in this situation, there was no significant effect on population. I wonder, though, can that result be extrapolated to other scenarios with perhaps, comparatively rarer species? If we are going out and sampling a vulnerable or an endangered species, is there a greater likelihood that there would be an impact on population?
Perhaps, and that's always a concern, and it's an issue that we need to take into account in terms of responsible collecting while we're practicing these techniques. I would say that collection of rare taxa is equally important to collecting specimens of more common taxa. And generally speaking, when you're out in the field sampling specimens, the comparatively rare species are caught much less often. And so really the specimens that are collected of those taxa are proportional to their relative abundance across the landscape. Well, one thing I would say in addition to that is that it should be kept in mind that most of the conservation efforts that are ongoing for comparatively rare taxa, and most of the wildlife management and regulations and identification of hotspots of biodiversity or regions of threatened biodiversity are all heavily reliant ultimately on specimen collections and utilizing the resources that are held within museum collections. And so really most of these specimens ultimately go on to contribute to beneficial conservation efforts and improving management of existing biodiversity into the future. That's
obviously a compelling case. Any last thoughts?
Yeah, I think I would just add that providing these specimens is a practice in foresight. And so much of the research that I do and that my colleagues, particularly evolutionary biologists, perform relies heavily on the efforts of others that have collected specimens through past decades. And sometimes we're utilizing specimens that are greater than 100 years old that are housed in these museum collections. And those specimens were originally collected by scientists who had no idea the potential value of those specimens into the future and had no inkling of the kinds of questions that we're asking right now. And so in terms of foresight, I think it's important for us to consider the potential for these specimens to be used by future generations of scientists who are asking questions that we can't foresee at this present time.
And that seems like a great place to leave it. Dr. Hope, thank you very much for joining me today.
My pleasure.
And that concludes this episode of BioScience Talks. Just a reminder, the journal BioScience is published by Oxford University Press on behalf of the American Institute of Biological Sciences and is made possible by the support of our members and donors. Thank you, and talk to you next time.